Next, we have Kaleb Goff, who will be discussing limited directional change in mountaintop plant communities over 19 years in the Sierra Nevada and Great Basin of the United States.
Awesome. Thanks so much. Let me just get into this interview here. Everybody seeing things okay?
Looks good.
Awesome. Thanks so much. Hi, everybody. My name is Kaleb Goff. I'm a fourth-year PhD candidate in Seema Sheth's Plant Evolutionary Ecology Lab here at NC State in Raleigh. Super grateful to be here and just super grateful to bring this research to such a broad audience. Really just respect and excited to be part of a Botanical Society of America symposium like this. I'm going to shift our focus a little bit more towards the community level, but still thinking about changes and thinking more about changes over time. And I'll start, let me just hide this video. And I'll just want to particularly just start by thinking about these long-term effects of climate on mountaintop plant communities. And so we'll go in that direction. Understanding, let me just turn this on actually. Understanding how species diversity varies across space is a foundational question in ecology. I think lots of us have wondered about that on walks and in talks and in our academic lives. Mountains, of course, and mountainous regions are these hotspots of biodiversity. So just including this great map here showing some really incredible sort of species richness numbers that we see kind of in mountains in the Andes and also mountains in Western North America. One of the most sort of famous biodiversity gradient, of course, is the latitudinal gradient. You can see that mountains sort of break away from this latitudinal gradient. Mountains have really steep climatic gradients over short spatial scales. Again, thinking about this question about variation of species diversity across space. They have lots of topographic complexity, and they have really rich aggregations. You know, the way to get species richness numbers or unique numbers of species that are this high in these sorts of regions is to have these really rich aggregations of small range species and have these centers of endemism. And this really is, I think, well described by the fact that in many of these mountain ranges and on many of these mountain peaks that we'll talk about today and that are kind of aggregated into this map, there are single site endemics. So for example, here or in the place that we'll be talking about today in eastern Nevada, in the Snake Range, there's a buckwheat there, Eriogonum holmgrenii, that we're looking at this summer that is just there and nowhere else on Earth. And so taken all together, I think these mountain places are really key places to understand not only the climatic controls on biodiversity, but also and spatial biodiversity gradients, but also on climate change impacts in these communities. There's sort of two models that have been proposed, two broad conceptual models for how climate change might be impacting mountain plants or mountain communities in general. One of them is this model that we call escalator to extinction, is proposed by Emma Maris and Mark Urban. And it's this, I think, really intuitive idea that with climate change, we might see species tracking, especially on top of mountains, we might see species tracking their climatic niches upwards, trying to remain where things are optimal. And for plant species or for species that occur on the very tops of these mountains, they might not have suitable habitat or they might not be able to disperse to suitable places to track their niches. And so we might see really high vulnerability, lots of extinctions and extirpations with rapid anthropogenic climate change. And then a contrasting sort of conceptual model for thinking about this is one that's been proposed by Christian Corner and others, which is to say that temperature or that these environmental gradients don't vary sort of cleanly across space. So for example, there's a picture of a mountain here, and then this is a thermal image of the same mountain here. And you can see that, you know, the coldest part is not the top and the warmest part is not the bottom. And in the context of climate change, you have species maybe that move from this warm patch here directly on contour, sort of left or right, rather than shifting up or down. They can find via this microclimatic heterogeneity and topographic, they can find these really small microhabitats. And so we might see really slow lag changes in this model. We can sort of combine these, and I think lags are going to be important. Jake Alexander created this really beautiful figure, I think, to talk about lags, whether community change happens quickly or slowly. There could be lags in different demographic processes, or different demographic processes could happen at differing rates. So for example, there might be lags related to extinction. In this case, some factors are proposed here, and this idea that you would have stronger lags, or things would take more time if you have a sharp arrow, and things would take less time, or you have shorter lags with the flat arrows and longer lags with the pointed arrows, and kind of 3 key stages for mountain plant communities changing at dispersal, or in dispersal, in establishment and extinction. And if we combine establishment and dispersal here, we can sort of apply into colonization to say that there might be some lags in colonization that might be changing communities at different rates, and there might be some lags in extinction that are changing communities at different rates. And that these can be, we can apply some of our most classic and most intuitive community measures to these to make some hypotheses. So we might expect that colonization lags are less than extinction lags. So colonization is happening quickly and extinction is happening slowly. That would lead us to have more species and more gains on mountain summits. We could expect the opposite, right? Which is that colonizations happen slowly, maybe because there are limited safe sites in these high elevation places, limited spaces to land, and that extinctions are slow, or that extinctions happen quickly, which might lend us richness decreases and more losses than gains. And then lastly, we might expect that there's no change in richness because the length of these lags is about the same. So to apply this and to look at some of these hypotheses, what we need is a whole bunch of data from a whole bunch of mountains. And luckily, there is this amazing international project started in Austria in 2001 by Georg Graber, an international effort called Gloria to monitor plant community change on alpine summits using this international and globally standardized protocol. And today, I'm going to talk about some of the results from 8 target regions, or 8 groups of peaks that are in Western North America. And inside of each of these 8 regions are 3 or 4 peaks. And each of the 3 or 4 peaks, or within the 3 or 4 peaks within a region, those peaks are laid out across an elevation gradient. So there are some peaks closer to the subalpine alpine ecotone and some peaks closer to the top. And I want to just emphasize that we're really talking about 10 meters vertical distance from these summits are the communities that we're going to be talking about today. So really thinking just about the summits. The protocol itself involves lots of early mornings and big hikes and lots of string. And we use string to set up these plots, 1 plot for each aspect, north, south, east, and west. And then those bisect 2 larger divisions, one for 5 meters and 10 meters. And so in each of those 8 plots, this is sort of what it looks like on the landscape here. We know all the species there, or the community composition, and then we know something about the categorical abundance on each peak and each year. And each of these peaks is surveyed every 5 years. And we're going to look at some data starting in 2004. I'll just say very briefly, so many people have made this possible, and these 8 target regions are sort of stewarded by a wonderful nonprofit agency called Gloria Great Basin. My advisor, Seema Sheth, is the science director for this awesome organization, and that this project happens thanks to volunteers that are willing to come out. So just wanted to show a couple of awesome photos from the work that we did this year. And if you're interested in this, I'll have my contact information at the end. Great. So if we look at some of the changes in climate, again, here are the 8 regions. This is what we're thinking. If we think about just what has happened over time in terms of changes in climate, we have seen increases. So on here you see mean annual air temperature just extracted from the climate layer and year. We've seen increases. The gray shows the time before the survey. So before the survey. So we're going to look at 2004 to 2022, but we might expect in these long-lived plants on mountaintops that the time before matters as well. So consistent increases. If you're familiar with this part of the world, you're familiar that precipitation is very variable there. So very variable precipitation in the decades preceding and during this study period. And then lastly, we bury these soil temperature devices on the summits. And what we've seen are some decreases. So this is mean annual soil temperature. And again, year, this just shows the period we have soil temperature logger data for, and we've actually seen decreases, and we think that's linked to decreases in snow cover. So what I'll show you here are the changes in species richness over time. So you'll see species richness on the y-axis and time on the x-axis. Each one of these triangles is a peak, and the peaks are laid out across an elevation gradient. So these dark peaks will be the highest, the lowest peaks. Sorry, the dark peaks will be the highest peaks, and the lighter peaks will be the dark peaks will be the lowest peaks, and the higher peaks will be lighter colored. So looking across all target regions, we saw no significant changes in species richness over time. A bit surprising to us, and I'll just foreshadow that there. There are some sort of not statistically significant, but some sort of positive slopes. And one of the questions I think we had from this was whether below these species richness changes, we were seeing just replacement of species turnover, or if we're seeing the same species every time we go there. So I'll show that here. And in this case, turnover is represented as the number of species gained plus the number of species lost, divided by the species richness between two sequential surveys, which gives us a sort of proportion of community change in each survey pair. And then I'll show the target regions here. What we saw, so each one of these small points is showing the difference in turnover between two sequential surveys, so sort of the raw data points. These colored dots show the averages for each of the 8 target regions. And then these long dark lines show the 95% confidence interval from a model. And then lastly, their average observed turnover was about 21%. So I think some would maybe say it's quite a lot of turnover. So what we did to try to figure out whether this was a lot of turnover or not is we created a null model, and the null model just randomly simulated species lists. So it took all the species that were seen in 2 sequential surveys, and then of the 2 surveys, it randomly sampled species until it reaches the species richness of a given survey. And then we're able to compare that. And then we show here the gray box is the 95% confidence interval from just random reshuffling of species. And the observed turnover falls pretty nicely inside of this null model or simulated turnover, which leads us to sort of conclude or to think that this turnover that we observed is similar to what we would expect turnover by random. Diving a little bit deeper into turnover, we partitioned the turnover by these functional groups people are familiar with: shrubs and trees, cushions, graminoids, and forbs. And what we found from doing this was that the greatest number of gains were in the forbs, and those included some of these rare and annual and ephemeral forbs. And then some of the greatest losses were in cushion plants, which is a bit surprising. And that overall this turnover was due to species chance colonization and extinction, and species blinking in and out rather than a directional change towards more species of a certain type. In summary, we're surprised to see limited plant community change across almost 2 decades and across across nearly 2 decades and across 29 summits. These results actually contrast what we've seen in other parts of the world where there are cooler and wetter climates. For example, showing on the right here, here's latitude and longitude, and this is just showing Northern Europe, right? So you can see Norway and Sweden and Finland here. And then each one of these boxes on the top, it shows the temperature change and then the species richness change over quite a long period. But they found really amazing increases in species richness on those summits. And it suggests this idea that climate change responses in the face of climate change, or community responses in the face of climate change might be heterogeneous and might depend on the sort of regional limitation on plant growth and performance. We're thinking quite a lot about future directions, and we're going to keep monitoring these summits as long as we can. This is the first chapter of my dissertation, and I'm really excited to think about more of the rates of change across the elevation gradients. So thinking down below the summits and between the subalpine and those alpine summits. We're thinking, of course, quite a lot more about functional traits or some ways to think about these species that moves away from their identity or tries to group them by their traits. And then thinking about changes in abundance. There's a little bit about change of abundance in this paper that you'll see when it's eventually out. And then lastly, we're working together to work on these North American and global syntheses to try to think more about these heterogeneous responses and seeing if we can sort of cluster these responses in different target regions over really broad continental scales. So in summary, thank you so much for your thoughts and attention. Chess Lab, the Chess Lab is looking for people who are interested in these types of projects, and I'd love to hear more about you. So feel free hear from you and hear your questions. So feel free to give me a shout or ask some questions. So thanks so much.